Okay, good afternoon, everyone. I hope you're doing well, and I hope you and your families are safe at this time. So, let's see. First off, please type yes into the chat window if you can hear me okay. Okay, great. Thank you very much. All right, gang. So, as seems to be happening in nearly all of my classes, folks are somewhat having a hard time attending, right? Attendance in classes is definitely dropping off, probably for a variety of reasons. So, try your best to stay focused and make the class a part of your continuing normal routine if you can. I know it's very challenging at this time. So, okay. Uh, what I want to do is just give you a chance to ask any questions you may have had about the recent lecture exam last week. As you know, the exams have been graded, and all you have to do is log into the class marker website in order to get your exam grade. Many of you did email me about concerns about, how the computer graded for things like misspelled words, or if you put in a plural version of a word, things like that. Please know that all of those things have been taken care of. I went back through and manually checked all of those things. So you can anticipate that those types of questions have been addressed, and please verify that by logging into your class marker account and checking your exam score. And if you still have questions after that, please contact me again through email, and I'll take a look at what's on your mind. Okay, so those questions have been dealt with. So at this time, what I'll do is go to the class marker website and very slowly scroll through the questions. And if there's something that you want to talk about or you have a question about, please type it into the chat window, or please unmute your microphone and say, hey, Dr. Allen, can we look at question 7 or something like that? Okay, so bear with me a moment. Okay, so I'm going to bring up the chat window here so that I can see your comments. And okay, I'm bringing up the class marker exam. Okay, so I don't want to go through every question blow by blow. We have lots to do today. So what I just want to do is show you a field of questions and give you an opportunity to interrupt me and to ask a question. So questions 1 through 3 are on the screen at this time. If you want to talk about anything, please type in the question number into the chat window. Okay, moving forward. The next few questions, questions 4 through 7, any of that you would like to ask about? Okay, moving on to questions 8 through 11, any of that you would like to ask about? Okay, moving on to questions 12 through 15. Anything here that you would like to go over? Okay, let's continue. How about questions 16 through 18? Okay, moving on to questions 19 through 21. Okay, moving on to questions 22 to 25. Okay, not hearing anything from you, I will continue. Moving on to questions 26 through 29. Anything you'd like to ask about? Okay. Moving on to questions 30 through 33. Any concerns there? Okay. Moving on to questions 34 through 37. Okay. Moving on to questions 38, 39, and 40. Okay. Question 41. That was our first essay question. And I think just about everybody got full credit on that. You did a nice job with your research for the Pearson syndrome. I thought many of you made good arguments on the ethical question, question C there, making an ethical argument in favor of or against terminating the pregnancy. I thought you might be interested to know that I actually counted your opinion and made a tally sheet up and let you know that, okay, so your class size is 9 people, so 9 people took the exam. 6 of you indicated that you thought aborting the pregnancy would be justified, and 3 of you indicated that you would be against aborting the pregnancy. So typically that's pretty, that's pretty much what we see on this question. The idea of abortion is a very hot topic that is contentious, and that spills into the science classroom as well. So, if we had a larger group, I would expect about 50% of students to be in favor of the abortion and 50% to be against the abortion. Okay, question 42. Any aspects of this that you would like to ask about? Okay, can we go over 42? Okay, well, okay. So let me go through each question here. Question 42, we have a plot of 9 different bacterial viruses, and we've plotted their melting temperature versus their percent GC content. So the percent GC content of the respective genomes. Question A, which viral DNA is most heat resistant? Well, it would be this guy up here, right? It would be the weird looking X, which is known as chi. That is the most heat resistant because it in fact has the highest percent GC content that causes it to have the actual highest melting temperature. So A is the weird X symbol here. B, what is the percent AT content of virus alpha? If you look at virus alpha and you look very carefully, its percent GC content is in fact 44%. So 100% minus 44% is 56%. That would be the answer for B, 56%. Letter C is the relationship between TM and percent GC directly or inversely proportional. It is directly proportional because you can see that as the melting temperature goes up, so does the percent GC, right? There's a positive linear relationship through the trend of the genomes here. So that is a direct relationship. Okay. Letter D, viruses T1 and T7 have the same percent GC content, but different melting temperatures. What could explain this? There's 2 different possibilities. Okay, it is very possible that T1 and T7 could have the same percent GC content, but literally have a different size genome. For example, 10% of 100 is 10, but 10% of 10 is 1. All right, so the percentage tells you, the percentage kinds of hides the fact that these viruses could actually have different sized genomes, different sized DNA molecules. So that's one possibility that could explain the different melting temperatures. The other possibility is the fact that even though these two viruses could have the same percent GC content, the position of the G's and C's along the DNA strand could be different, which would allow the secondary structure of the virus to be different. So what I'm suggesting is that the DNA can fold into different shapes, allowing one version to be more heat resistant than the other. Okay. Questions about that? Okay. Letter E, which virus has the best chance of expressing its gene when its cell is frozen in the permafrost of Siberia? Well, I simply wanted you to see that alpha has the lowest melting temperature. It has the lowest melting temperature, which means it's going to take the least heat energy to open up the helix of the DNA to express its genes. Okay. Letter F, what is the percent purine content of virus MU1, whose percent GC content is 51? Well, if this virus has percent GC content of 51, then half of that is 25.5. That means the amount of G's is 25.5, which means the amount of C's is 25.5. So that means the remaining content of percent AT, which would be what we're after here, is going to be 49, and 49 divided by 2 is 24.5. So A has 24.5 and T has 24.5. So if we're looking for percent purines, we need to add 25.5 and 24.5 to get 50%. Gang, does that make sense? Right, we needed to calculate the amount that's due to the G's, which is 25.5, and the amount that's due to the A's, which is 24.5. So the purines are A and G nucleotides, so we add 25.5 and 24.5 for a total of 50%. Okay, how about question 43? Anything there you want to ask about? Okay, I will say that on question 43, section F, the very last question here was most commonly missed. And I do believe, although I'm going off by memory here, I do believe that the only person in your entire class to get that question right was Sean. So good job, Sean. Everybody give him a virtual applause. Sean got question F right, and I'm pretty sure everybody else got it wrong. I'm pretty sure that's true. Okay. All right. So F is bottom to top. Question 44. Any questions about question 44? Okay, not seeing anything. I will move to question 45 here, which was the last one. Okay, this one was a trouble spot for many of you. In fact, again, going from memory, I think the only person in your group to get question A correct was Ian. So let's give Ian a virtual applause. Ian got question A correct. Convert the coding DNA sequence into a messenger RNA sequence. This is the coding DNA sequence. That means it's going to have the same sequence as the messenger RNA, except you got to put U's everywhere you have T's. So you literally would have written down on your exam or typed it into the computer, GU, CCC, AA, CC, AUG, and so on the whole way through here. The answer to B is nucleotide positions 10, 11, and 12. The answer to C is nucleotides 41, 42, and 43. The answer to D is the following sequence of amino acids: Met, Pro, 3 as in threonine, ASP, ASP, Lu, Pro, ALA, SER, SER, Glu, ASP, ALA, Gly, GLY, and Pro. The answer to D is Met, Arg, ALA, and Glyn, GLN. Letter E, are the two questions you created in D and E in the same reading frame? Well, they would not be, because there's no overlap of their amino acids, so they are not. And the last question, and again, I apologize, I had 2 letter D's in here. That's just a typo, but you all handled that just fine. Letter F suggests a reason why Mother Nature could use the same gene to produce 2 different proteins. Well, typically the argument that's given is that, you know, if you reuse the same information 2 different ways, that allows the overall genome of the organism to be smaller, which means the cell doesn't have to spend even more energy maintaining that genome. Okay. All right, gang. Do you have any questions about this exam? All right. Fair enough, gang. Let me take that off the screen and stop sharing with that. Okay. Oh, John, you wanted to know about where the start codon is. Okay, let me bring that back up here. John, the start codon for the last question is simply going to be where that first AUG is. Okay, right there it is. Okay, any other questions? Okay, so gang, the class average on the exam was a 75%, which is actually higher than your first exam. So that means the trend in your performance is improving, and that's always a good sign. Okay, so, you know, take some heart in that you are collectively getting stronger. Okay. The final exam, which is only, you know, about 5 weeks away at this point, you will have 2 hours to take the exam. Okay, so I know some of you are concerned about the amount of time you're given to take the exam, but, you know, I always have to balance how much time to give you, how much stress I want to give you. And so I did want to give you some stress on this exam. So for the final exam, you will have 2 hours to take that test. Okay, do you have any questions about the exam before we move to new topics? Okay, gang. All right, so let me know if there's something bothering you. Shoot me an email. Several of you did shoot me an email, and I didn't necessarily respond because I had already fixed the problem. So if you do have new or continuing concerns, please send me an email. Okay. So I thought at this point I would quickly go back over with you how to calculate your course grade. I know that, you know, I believe at this point that the last day that you can technically withdraw from the course is the end of April. And, you know, I personally don't see any reason why anybody needs to withdraw, but that is a decision that you have to make for yourself. Okay, so I do want to show you how to calculate your course grade, or at least estimate it at this time. So bear with me as I go back to Microsoft Excel and show you how to do that. Okay, so to calculate your course grade, okay, and I'm going to bring Excel up, bring the chat window back up. What we're going to do is make a quick matrix in the spreadsheet. And of course, you can do this at home with your own scores, your own grades. I'm just going to do a hypothetical example of how you could calculate a grade in the course. So in your course, There are 3 lecture exams, lecture exam 1, 2, and 3. Okay. You also have your programming. Well, let's do it this way. You also have your lab work, which, you know, at this point, most of the lab content's going to be awarded to you automatically because of our emergency. We have the textbook homework. We have the programming work you do with Mathematica. We have the 2 literature reviews, literature review 1 and literature review 2. Okay. And all of these things will be combined at the end of the semester to give you your course grade. So, okay. So again, you could do this calculation with your own scores, but I'll just use hypothetical numbers at this time. So for the first lecture exam, the class average was a 70%. So you put that in as a decimal. Class average on this most recent lecture exam was a 75%. So again, put that in as a decimal. Lecture exam 3 is your final exam, and we don't know what that's going to be yet. So I'm going to leave it momentarily empty. For the lab, okay, for the lab, what I'll say is this, is that if you completed all your lab write-ups, successfully before we went to our emergency break, then the amount of credit you would get in the course for your lab work would be a total of 12%. So I'm going to put 12% in the weighted score column. Okay, for textbook, if you have received all credit for your textbook homework, you've done all of it, there's been no major problems, then you would get 14% of your course grade through that. For programming, if you've been keeping up with your programming, you haven't been missing questions and things like this, you would get 14% from that. Where am I getting these percentages? I'm getting it from the very front of your syllabus, the front page of your syllabus. Okay.
What's the programming exactly?
That's the Mathematica work you're doing, where you have those little programming activities every week.
Okay.
Okay, so you're getting course credit for learning Mathematica as well as doing the textbook genetics homework problems. Now we have 2 literature reviews. You've already finished your first literature review, and you know most people earned an A of some kind on that, and the total percentage you could get for each one of those is 3%. Okay, so if you got 100% on the first literature review, it's going to count as 3% of your weighted score. And you have your next literature review coming up due this coming Tuesday, a week from today. So I'll talk about that in just a moment. Okay, but if you get everything perfect on that one, you'll also get 3%. Okay. So the very last thing is this darn lecture exam 3, right? This is your final exam. It's at the end of the semester. It will be on chapters 11, 8, 12, 13, and 14. Okay. And so you need to ask yourself how well you think you can do on your last exam. So you kind of have to make an estimate. You have to make a guesstimate of what you think you could get. Okay, so everything in yellow is an estimate. All right. Obviously, some of these things, textbook and programming, are also estimates based on your personal situation. Okay, but let's say for the third lecture exam, let's say that the class average is an 82%. Okay, let's do that. Now, the first lecture exam is weighted as 16% of your course grade. I'm taking that from your syllabus. Your second lecture exam is 19% of your course grade, and your final exam is also 19% of your course grade. To get the weighted scores for your exams, you take the score and you multiply by the weight. And again, the weights are coming off your syllabus. So I'm going to do that for all of your lecture exams. And then once you have all the data in the weighted score column, to get your final grade, you simply add them all together. So I'm going to add everything in column D together. And at least in this for this hypothetical student is going to finish the semester with an 87%, which is a nice solid B. Okay. So that's the process that I will go through when I calculate your grade at the end of the semester. Do you have any questions about this? Okay. Well, let me know if you do have a question. Hopefully you can do this for yourself and it'll give you some confidence where you actually stand in the class. If you do it and you have questions, or if you're having trouble doing it, shoot me an email and we'll work through the calculation together so that you have some confidence where you stand. But I think, it's certainly true that not everybody's going to get an A, but it's also true that I think everybody's certainly going to get a respectable exam, a respectable grade, as long as there's no major snags moving through the next 4 weeks of the course. Okay. All right. Any questions? All right. All right. I'm going to stop that. Okay, gang. So, yes, let me remind you that Literature Review 2 is due a week from today. You all received that document through email weeks ago, okay? It's an article called Saving Science that you have to read, and then there's a Word document with many questions that you have to answer. Since we are separated from each other, please type your answers into the Word document and save that typed version of your answers so that you can email it to me a week from today, because what I'll have to do is read your electronic documents and grade them on the computer. So please answer on the electronic Word document, save that, send it to me next Tuesday for completion. Okay, a couple other things. Let me see here. I'm trying to think back to some things I was noticing today. Jose, I sent you an email. I think you sent me the wrong assignment, so please check your email. Hayden, I also sent you an email about your homework. Please check that today. And let's see, Kevin's not online, is he? But okay. Sean, I have not received your homework that was due last Thursday. It's not in my junk folder or my inbox, so please send that again to me. Okay. All right, gang, that's a lot of business item stuff there. Do you have any questions before we continue? All right. All right, gang, so let's get back into it. My goal is to finish chapter 11 today and be on to chapter 8 next class period. That's my hope. Let's see what we can do here to make that a reality. So today's continuing lecture on chapter 11, remember we were talking about different kinds of mutations that can be created inside cells. We also talked a little bit about mutation rate, and I gave you that little puzzle of starting with one cell and allowing that to reproduce to 1 billion cells. How many mutations would we expect at the lac locus? We did that at the end of last class. So what we're going to do next is go into different mechanisms. Well, what we're going to do is describe different ways that mutations can occur in cells. We're not really going to go into mechanisms very detailed, because honestly, even a single mechanism, I could lecture an entire hour and 15 minutes on, and we wouldn't get through everything. So we're just going to describe some of these, and I'm going to show you a variety of videos today about many of these topics. So this is going to be a real show and tell lab where I'm talking a little bit and showing you videos at the same time. Okay, so let's continue here. Biology 215, Chapter 11, continued. Okay, today's date is the 14th. All right, so we're going to talk about mechanisms of mutation. How do mutations occur inside cells? Okay, well, that's big and complicated because there's a lot of different things that can monkey around with the DNA sequence inside bacterial cells, inside your cells, animal cells, lots of different things that can go haywire. Okay, you know, it's very, very difficult to get an accurate quantitative number on the number of mutations that our cells experience on a daily basis. But I think a ballpark estimate of that most scientists are comfortable with at this time is that probably every cell of our body on average in any given day experiences tens of thousands of injuries to the DNA. Tens of thousands of injuries, tens of thousands of mutational events to the DNA that have to be repaired successfully, or the cell may die, or the cell might grow in a way that promotes disease. And obviously that's something we really don't want. Okay, so number one here. Okay, number one here. Okay, so what I'm going to be doing is going through pages 309, 310, 311. Okay, we're going to be taking a survey of these things. The first thing is that DNA polymerase can simply make mistakes, right? It is, after all, a biological machine, and so it should not be a surprise to you that sometimes an enzyme like DNA polymerase makes errors. Okay, typically these mistakes occur when one strand of the DNA actually forms a loop. Okay, actually, because of the nucleotide sequence can form a loop that will confuse the DNA polymerase enzyme, causing it to insert wrong nucleotides. Sometimes because of those loops and other localized structure in the DNA helix, sometimes the DNA polymerase will actually slip. Like you slip on a piece of ice on the ground, you slip. Sometimes DNA polymerase can actually slip forward or slip backwards, or it can stutter. All of these things can cause a mutational event to occur. These mistakes can occur when one strand of the DNA inappropriately loops out, or when DNA polymerase slips, so to speak, slips or stutters. Okay, and those slipping and stuttering motions would be induced by localized malformations in the DNA structure. Okay. It really stinks that we're not able to do our lab content together anymore, because I was going to do a forensic PCR lab with you. You might have heard of PCR, maybe you've even done PCR, it's polymerase chain reaction, but we were going to use it in a forensic setting where we had collected evidence at a mock crime scene and used, you know, forensic analysis of our PCR results to determine who committed the crime. We were going to do this. We can't do it anymore. But at any rate, in that write-up of the forensic PCR, there's a whole section on what is called VNTRs, variable number of tandem repeats. Okay, these are areas of the genome, areas of the DNA where you have repeated segments of nucleotides that are particularly prone to mutational events, because repeated sequence of nucleotides seems to give DNA polymerase problems with slipping along the DNA template. So let me put that into your notes. Certain areas of DNA containing tandemly repeated sequences called VNTR, short for variable number of tandem repeats, excuse me, variable number of tandem repeats seem to be prone to a phenomenon called replication slippage. Okay. This can lead to both insertions and deletions in the DNA. Okay. Please feel free to stop me at any moment. Type question into the chat window if you have it. At this time, I'm going to show you a YouTube video, which is a little quick video that gives you a basic idea of how DNA polymerase slipping can actually occur. Okay, so if you haven't finished your note taking, I will be coming back to the Word document, don't worry. But let me show you this video on DNA slipping. Okay. All right, so here's a video called Slip Strand Mispairing. It explains why DNA polymerase can actually slip and what has to happen for the polymerase to actually correct that issue. Okay, so here we go.
Slip-strand mispairing occurs when a nucleotide sequence is present in more than one copy in a chromosome, and the copies on homologous chromosomes pair out of register. A loop is formed in one strand of the DNA as the mismatching occurs. This loop is sometimes excised by the cell's repair enzymes, producing a short deletion. Some chemicals specifically promote deletions and frame shift mutations by stabilizing the loops produced during slip-strand mispairing, thus increasing the time the loops are vulnerable to excision. Many of these deletions start in the middle of a codon, thereby altering the reading frame. These so-called frame shift mutations cause the information in the gene to be read in the wrong 3 base groupings, distorting the genetic message.
Okay, folks, I think that's it for that video. But hopefully what you were able to see from the video is that when you have tandem repeats of DNA, sometimes the wrong repeat on one strand inappropriately base pairs with the opposing strand's repeat. They get out of register with respect to each other. That can happen naturally through thermodynamic fluctuations of heat energy in the cell. It can also happen because a chemical of some kind is interacting with the DNA and distorting its structure. Okay. Do you have any questions about that? Okay. All right, so let's continue here. That's one possibility of what can cause DNA mutation. Another possibility for DNA mutation is that the DNA nucleotides can experience what's called as a tautomeric shift. Okay. Okay, and for this, I'll be using figure 2 on page 309. Figure 2, page 309. Okay, and so first off, let's define what a tautomer is. Okay. Okay, a tautomer is an isomer. So you may remember back from your chemistry days, okay, a tautomer is a form of isomerism, okay, where we understand that an isomer Isomers are molecules that have the same number of atoms, but in some scenarios, isomers have the atoms arranged differently. In tautomerism, the atoms are not arranged differently, but the bonding pattern, the bonds between the atoms are different. Okay, a form of isomerism in which molecules have the same atoms in the same spatial arrangement, but the bonds between the atoms are different. Okay? So as an example, let me pull up a little PowerPoint here. Okay? So once again, a tautomer is a form of isomerism in which molecules have the same atoms in the same spatial arrangement, same geometric arrangement, but the bonds between the atoms are different. And that's because electrons and protons are kind of moving about in the molecule. So we won't worry too much about the chemistry, but have a look here on your screen. Okay, this is actually figure 2 on page 309 of your textbook. And what you'll notice here is what I want to highlight for you, and please watch my cursor on the screen. Notice on this thymine, which the book is telling you is in the keto version. Don't worry what that really means. I'm not going to pound you on the chemistry, but take a look here. Where my cursor is, we have an oxygen, followed by a carbon, followed by a nitrogen, followed by a carbon, followed by an oxygen. Down on the bottom, we have the exact same arrangement of atoms, right? I have an oxygen here, just like I have an oxygen up here. Next, I have a carbon, a nitrogen, a carbon, and an oxygen. So the arrangement of the atoms is identical. That is, there is no difference here. But where the difference is, take note here where my cursor is. On the top version of thymine, notice between this carbon and this oxygen, I have a double bond. But on the bottom version of thymine, I have a single covalent bond between carbon and oxygen. And so I have a single bond here and I have a double bond here. That means that I can form on the top molecule, I can form a hydrogen bond. The dashed lines are hydrogen bonds with the next nucleotide on the top sequence. And down here, I'm going to have a covalent bond with a hydrogen. Okay, that's the tautomerism we are talking about. The atoms are the same, but the bonding pattern is different. Okay, the bonding pattern is different. Okay, so as an example, when you have a thymine that's in this keto arrangement, where keto is sort of defined by this double bond, that allows thymine to base pair normally with adenine. And you're used to that, right? You're used to that. ATs base pair together, that's normal. That's normal DNA. But when thymine takes a tautomeric shift into an alternative form called the enol form, where we take the double bond and we transition to a single bond here and a double bond here at the nitrogen, then all of a sudden thymine starts base pairing with G, guanine, which is highly irregular and highly unusual, and we don't teach it that way in classes, do we? But the reality of it is this is happening in your cells probably millions of times a day across the cells of your body, okay? Same thing's going to happen with cytosine, right? Cytosine has a certain form called amino, and it has nitrogen, carbon, oxygen. Same on the bottom, but take a look. When this thing shifts from the amino form to the imino form, A to I, notice that double bond up here notice where that single bond where my cursor is in fact converted to a double bond. That allows cytosine, which normally base pairs with G, to all of a sudden start base pairing with an A. Very irregular, at least in terms of basic biology dogma. So these are examples of tautomeric shifts. Okay, any questions? All right. So, okay, these tautomeric shifts occur due to fluctuations in heat energy inside cells, right? Our cells typically are maintained at about 37 degrees Celsius, but if you start going up to 37.1, 37.2, if there's even a minor fluctuation of 1/10 of a degree Celsius, then some of the nucleotides in our DNA are going to start experiencing a tautomeric shift into the alternative form and causing alternative base pairing possibilities to occur. That can lead to permanent mutations inside cells. Okay. Any questions? Okay. All righty. Number 3. Okay, we're talking about ways that mutations occur inside cells. Well, there's the possibility of different forms of nitrogen-base damage. Okay, we know that nitrogen bases are one of the 3 primary components of the nucleotide. And there's different ways that those nitrogen bases can in fact be damaged. Okay, so I'm talking about things like you might see in figure 4, page 310. All right, so nitrogen base damage can actually cause mutations. There's 2 typical forms of this that can occur. First, we have depurination. Okay, depurination is the removal of one of the purine nitrogenous bases. So a removal of an adenine or guanine nitrogen base. Okay. As far as biologists are aware, this seems to be fairly common in cells because depurination as a process can occur because the DNA is interacting with molecules that are inside the nucleus. That would include things like hormones that enter the cell, like estrogen. Estrogen is known to do things to DNA, to depurinate different locations of DNA, typically caused by molecules like hormones interacting with DNA in the nucleus. Okay, examples of molecules that interact with DNA include estrogen, and ironically enough, and paradoxically enough, and perhaps even surprisingly enough, sometimes even just having the adenine or guanine interact with water can cause the depurination. Okay. So depurination is typically caused by molecules like hormones that interact with the DNA. That would include estrogen, so hormones, and even water alone all by itself can sometimes promote the depurination event. I'll come back to my notes, but let's take a look here on the screen. Okay, so here we have an example of a typical depurination event where literally that guanine nitrogenous base on the left side of the molecule here is cleaved off and it is no longer attached to that sugar. Okay, that's what I literally mean by a depurination, where the nitrogen base is literally chemically detached from the rest of the nucleotide. That is depurination. Okay. Any questions? As I mentioned earlier, it is very difficult to get accurate quantitative numbers on how frequently these things occur, but at least some reasonable ballpark estimates on depurination is that probably every cell of our body probably experiences on average about 5,000 depurination events every day. Okay, I mean, You think about that, 5,000 depurination events in every one of maybe 75 trillion human cells that make up our body. You can begin to see just how many mutational events that our cells of our entire body have to deal with every day. And when you start thinking about it in that way, you start realizing that, my God, it's amazing we're even alive at all. Okay. The second form of nitrogen-based damage that can happen is deamination. Okay, deamination is the removal of an amine group from nucleotides like cytosine or adenine. So the conversion, deamination is the conversion of cytosine to uracil, or the conversion of adenine to an alternative nitrogen base that you may not have heard of before, called hypoxanthine. Okay, don't worry about the chemistry, I promise. I'm not going to beat you up with it. If I am going to beat you up with it, I'll give you warning before your final exam. Okay? But as we go to the PowerPoint presentation again, let me just show you what I'm talking about, which is an example of figure 4 out of your textbook, right? Hold on a sec. Okay, here in the top left we have cytosine, and you notice it has the NH2 group, which would have been derived originally from ammonia, which is why this is called an amine group. Notice that when this is cleaved off and we form a double bond with oxygen, we can now base pair. When this is cleaved off and we form a double bond, the cytosine itself is literally converted into uracil. Okay, that uracil then can base pair with the adenine, but obviously that would create a mutation in your DNA. If you're converting cytosine, which should be base pairing with G, you convert it to a uracil, which then base pairs with an A, and all of a sudden we have a mutational event in the DNA itself. Same thing is going to happen with adenine. If you take the amine group and you rip it off and you form a double bond with that carbon with an oxygen from a water, let's say, you convert the adenine to hypoxanthine, which then can base pair with cytosine. So adenine, which normally base pairs with T on the opposing strand, will convert to something that will now base pair with C. So again, we have a mutation in the DNA sequence. Okay, so that's a very primitive overview of the chemistry that's happening there. Okay, the deamination that occurs in cells can be spontaneous in the sense that it's not predictable, but deamination is also known to be catalyzed by specific enzymes in the cell that are trying to manipulate, molecules in the localized environment. Can be caused by specific enzymes in the nucleus, but is also known to happen spontaneously. All right, I'm going to keep rolling, gang, because there's so much I want to get into your notes. Please feel free to interrupt me. Please type questions into the chat window if you have them. All right, so the third form of mutational mechanism was nitrogen-based damage. Okay, there's other things that your book does mention. It does mention oxidative damage. Okay, in this circumstance, what we're talking about is the production of toxic forms of oxygen, reactive oxygen species that are typically buildup products, waste products from aerobic cellular respiration. Those things can also not just damage DNA, but damage proteins, lipids, carbohydrates as well. Okay, so oxidative damage is caused by the toxic forms of reactive oxygen that accumulate during aerobic cellular respiration. Now, obviously, aerobic cellular respiration is happening predominantly in the mitochondrion, but that does explain why mitochondrial DNA experiences a higher rate of mutation than nuclear DNA. But even so, some of those reactive oxygen species can leave the mitochondria and float into the nucleus and cause harm there as well. Your book mentions that over 100 different chemical modifications that are due to oxidative damage have been cataloged and surveyed by scientists in the scientific community. Over 100 different chemical modifications due to oxidative damage are known. Okay, next we have transposable elements, right? Transposons, jumping genes. Well, these things also can cause havoc with our DNA. Okay. I'll try to slow down a little bit if I'm typing too fast. Many of the transposons that we are aware of, many of the transposable elements that we are scientifically aware of, do seem to jump around the genome fairly randomly. At least there's no obvious sequence prediction capability to where they move. In other words, there's no commonality to the target sequence that they hop into. That's not to say there isn't a three-dimensional structure sequence that they're hopping into, but we're just not that sophisticated in predicting things yet. Okay. Many, if not most, transposons excise mean remove themselves from the DNA and randomly insert themselves throughout a cell's genome. Okay, obviously that could have drastic consequences for disrupting of genes that are important to the health and viability of a cell. So I do have a video on that. Let me show you the basic mechanism that we currently understand for how transposable elements move about the genome. Okay, one moment. All right, let's see. I think I had, there it is, okay. Mechanism of transposition. Here we go, gang. Let's take a look at this. This is how jumping genes move around.
Transposable elements are segments of DNA that move from one genomic location to another. The simplest transposable elements are insertion sequences, or IS elements. An IS is a short sequence of DNA carrying only the genes needed for transposition and bounded at both ends by sequences of nucleotides in reversed orientations known as inverted repeats. Between the inverted repeats is the gene for transposase, which recognizes the ends of the IS. Because the enzyme is not specific for a particular sequence on the genome, transposable elements appear to move to random destinations. Transposable elements can also contain genes other than transposase. For example, some carry antibiotic resistance genes or toxin genes. These elements are called composite transposons. Composite transposons consist of a central region containing the extra genes flanked on both sides by IS elements. When a transposon inserts at a target site, the target sequence is duplicated so that short direct sequence repeats flank the transposon's terminal inverted repeats. Typically, the transposon remains at the parental site, while a replicated copy inserts at the target DNA. This is called replicative transposition. In this example, a transposon moves from one plasmid to another, leaving a copy behind.
Okay, gang. So I don't want to overemphasize or belittle the complexity of the mechanisms we're talking about here. You understand that everything that you're getting like a minute and a half video on could very easily be the topic of somebody's PhD dissertation in graduate school. I mean, these processes are that complicated and have to be studied in such excruciating detail that you could definitely go to graduate school and earn a PhD on the molecular genetics of transposon mobility. That is something you could do if you were so motivated to do it. So these things are very, very complicated, and we're just hinting at their existence at this time. Do you have any questions? I know it can be overwhelming when you get all these things thrown at you so rapidly. Okay, let's see here. So number 5, the jumping genes. Those definitely can cause mutations. And you know, jumping genes share an evolutionary history with viruses. And both transposons and viruses definitely are forces in Mother Nature that have influenced the evolution of life on this planet, including the evolution of Homo sapiens. Okay, number 6. We'll just put a couple things into your notes here, and we'll call this topic put for the day. Well, obviously, these things that we've listed can all cause mutations in the DNA, but certainly can just certain special chemicals, right? Chemicals, there are certain chemicals that are known to cause mutation. One such chemical would be the nitrogen base analogs that you see on figure 5, page 312. Okay, figure 5, page 312. Also figure 6, page 312. Okay, so let me list a couple of these things down. There are nitrogen base analogs, meaning things that are very similar and can be used as a substitute. Okay, that would be figure 5 on page 312. Okay, you can notice how a substance called 5-bromouracil, right up at the top of figure 5, 5-bromouracil can experience a tautomeric shift. Okay, so its bonding pattern shifts, causing it to base pair with guanine, which then leads to a mutation during the subsequent round of DNA replication. Notice how 5-bromouracil can experience a tautomeric shift, allowing it to base pair with guanine, leading to mutation during DNA replication. We also have other things that are listed. We have what are called alkylating agents. Okay, alkylating agents would include ethyl methane sulfonate, which you see in figure 6. Okay, alkylating agents like EMS, like ethyl methane sulfonate. Okay, like ethyl methane sulfonate, can convert a nitrogen base to a derivative that can base pair with something other than what was originally intended. So alkylating agents like EMS can convert the nitrogen base of a nucleotide to a derivative, allowing it to base pair with a non-canonical, with an abnormal nitrogen base partner. Okay. By the way, some of you may be aware of, you know, if you're a history buff, if you've ever studied World War One, you know, World War One in some ways was more horrific than World War Two because it was during World War One where civilization became advanced enough that we could deliver gas weapons, chemical warfare weapons to the enemy, but unfortunately, a lot of times the chemical fumes would simply float back onto our own troops. But long story short, chemical warfare, gas warfare included a substance that you may have heard called mustard gas. Mustard gas is an alkylating agent, and this is how mustard gas mutates the DNA. I say it's mustard gas of World War One fame, although it's not a good kind of fame. It was a very hard time of our humanity. Okay, one more thing that I'm going to put down in this category here is intercalating agents. Okay, I don't think your book gives you an actual picture of these things, but okay, so when you intercalate DNA, if you think about DNA as a ladder, and you have, you know, the left side of the ladder, the right side of the ladder, and the rungs connecting the left and right sides, and these rungs are stacked on top of each other. Well, if you intercalate that structure, you put a molecule, you put a chemical in between the rungs of the ladder. You put a molecule in between the nitrogen base pair rungs of the DNA ladder. Intercalating agents like ethidium bromide can insert in between the nitrogen base pair rungs of the DNA ladder and disrupt DNA replication. Okay, it can disrupt DNA replication. It can disrupt transcription itself, and it can also interrupt DNA repair. Okay. Let me finish this topic, gang, so we can move on to some repair mechanisms. Okay, so far all I've covered is different ways that different sources, different triggers in the environment can actually screw up our DNA. All right, so number 7 here is ultraviolet light, ultraviolet radiation. I'm talking about sunlight, right? Sunshine, people being worried about skin cancer. UV light can promote a form of DNA mutation called thymine dimers. Okay, the thymine dimers are shown to you in figure 8 on page 313. Figure 8, page 313. And when you get a thymine dimer, you have 2 thymine nitrogen bases on the same strand of the DNA helix that form an improper covalent bond in between them. Okay, and that's what figure 8 is trying to show you. Notice that you have 2 thymidine nitrogen bases there, and what's happening is that covalent bonds are forming between their atoms. That should not be happening. Okay. All right. We'll talk about some of the repair processes in just a moment. The last thing I'll put into your notes is ionizing radiation. Okay. When we talk about ionizing radiation, we're talking about things like that are fairly energetic, different forms of radiation that are pretty energetic, such as X-rays, right? If you've ever had an X-ray of your jaw at the dentist to check for cavities or anything, if you ever had a jaw for, if you ever had an X-ray for a broken bone, X-rays are a form of ionizing radiation. So X-rays, gamma rays. We have any superhero geeks out here for the Incredible Hulk? Okay, gamma rays are a form of ionizing radiation, and cosmic rays. Okay, cosmic rays from the background of our universe are these forms of radiation are strong enough to actually penetrate the outer layer of our tissue, get inside our body into the cells, and actually break chemical bonds and molecules, not just DNA, but protein, carbohydrates, and lipids too. So X-rays, gamma rays, and cosmic rays are all energetic enough to penetrate the cells of our body and alter the chemical bonds in a variety of organic molecules, including DNA, right? That's why even though it's important sometimes to get X-rays at the doctor or the dentist for medicinal purposes, we don't want to overdo it, right? Because X-rays can cause serious mutation that if the repair mechanisms in the cell can't repair, well, that could cause a problem. All right. Gang, do you have any questions about the mechanisms that can screw up our DNA? When you look about the number of different ways that the environment of our body and cells are in that can actually damage our DNA, it truly is a miracle that, most of us are blessed to be able to live 60, 70, 80, maybe 90 years of life before we kind of implode upon ourselves. Any questions? Okay. I hope you're not unconscious on your sofa. All right. Let's see here. The next and final topic of this chapter, which it looks like I'm not going to get it done, but I got to do it with you, is DNA repair mechanisms. Okay, so we've now cataloged a variety of ways that your DNA can be mutated and damaged. But thankfully, Mother Nature is pretty intelligent, and she has created a variety of mechanisms that can repair damage to DNA. And a lot of times when we get disease in our body, it's because the repair mechanisms are broken. The repair mechanisms have failed to correct an error of some kind. So let's talk about the DNA repair systems. I know I'm getting short on time. Please bear with me here a little bit. Okay. So let's see here. I'm going to material that's now on page 316, 317, 318, 319. We won't get it done today, but hopefully within a few minutes of next class, we will get it done. Okay. So, all right, we've cataloged a variety of ways that our DNA can be damaged. What ways do cells have to fix that damage? That's what we need to talk about. Fortunately, DNA polymerase enzymes usually have what's called a proofreading activity, which I know many of you learned about, studied about, and read about for your previous exam when you talked about DNA replication. Some of you focused on the idea that, in fact, DNA polymerase often has what's called a 3' to 5' exonuclease activity. So DNA polymerase enzymes usually have a 3' to 5' exonuclease activity, which allows the enzyme to go in reverse. It's like putting your car's transmission in reverse and backing up a little bit so you can redo a mistake and correct it. Allows the enzyme to go in reverse to cut out any incorrectly placed nucleotides. Okay, gang, I'm going to show you a quick video on this, and then we will wrap it up today. So please bear with me here. I'm going to show you a quick video on this. Oh my goodness. Let's see. Okay, here's a video. By the way, I'll be sending you a lot of links to these videos for your own personal study time. Proofreading of DNA polymerase. Here we go, gang. Let's take a look at this.
Although mistakes can happen during DNA replication, they are extraordinarily rare. A key reason for this is the proofreading function of DNA polymerase. Roughly 1 out of every 100,000 to 1 million bases attached to the 3' end of a growing DNA chain will be a mismatch, improperly base pairing with the complementary strand. When such a mismatch occurs, DNA chain elongation generally pauses. This pause allows spontaneous melting of the end of the DNA strand being synthesized, freeing the 3' end with the mismatched base to enter a 3' exonuclease site on the DNA polymerase. The exonuclease site catalyzes the removal of several nucleotides from the 3' end of the growing strand. Only the removal of the nucleotide carrying the mismatched base is shown here, though additional nucleotides would actually be removed. Following removal in the 3' to 5' direction, DNA polymerase can then resume its synthesis activity in the 5' to 3' direction. The proofreading function reduces the overall error rate to as low as about 1 in 100 million for DNA polymerase 3. Although mistakes can...
Okay, gang. Let me go back to the Word document here. All right, so first off, let me say, if you're feeling overwhelmed by this content, don't be, because again, each individual statement we made today was somebody's PhD dissertation a long time ago. These things are complicated, and while you have to know about them, and you have to be able to answer vocabulary type questions on a future exam, I'm not going to ask you things about the intimate chemistry of tautomerism on the exam. So, put it into perspective exactly what you will be expected to know. Of course, do your homework and continue to learn that way. So, do you have any questions before we stop for this afternoon? Before you take off, let me put it into your mind that I really, really need to cover ground next class period. We're getting a little bit behind. And so next class period, I will continue with these DNA repair systems, and then I will start chapter 8. If I'm running out of time in chapter 8 for next class, I may continue into what would normally be our lab time together on Thursday, because I don't want to get too far behind here. So please at least be open to the possibility of sticking around a little bit extra time on Thursday if we need it to make sure chapter 8 can be covered properly. Do you have any questions? Okay, I'm going to stop sharing. I will have my office hour this afternoon. If any of you need to stick around, want to chat with me, or have a question of some kind, I'll be back online here at Zoom in 15 minutes, 2:05. Be happy to help you with anything. So if I don't see you until Thursday, please stay well and take care of yourselves. So I will see you soon. Have a good afternoon.